All right, this is Microbiology Lecture 7. It's on viruses. The general characteristics of viruses are that they are extremely simple, stripped down, the barest, most stripped down versions of something that can be considered to be alive. They, in fact, are so stripped down to the bare essentials that they cannot reproduce on their own. They can't reproduce themselves on their own. They have to infect a host cell. And so they always require the host cell's machinery in order to replicate. Essentially, they are obligate intracellular parasites. The viruses, there are different groups for each different major organism out there. There are animal viruses, there are plant viruses, fungal viruses, viruses that infect protists or protozoa, and there are bacterial viruses. They're extremely small, anywhere from, they range in size. The largest ones are 1,000 nanometers, but they can be down to, and they usually are closer to the lower end, they're anywhere from 20 to 1,000 nanometers in size. 1,000 nanometers equals 1 micrometer, and a red blood cell typically has a diameter of about 8 to 10 microns, or micrometer. Micron is a micrometer. So you can see that they are extremely small. They all contain two things. One is nucleic acids, which code for themselves. And the nucleic acid can be either DNA or RNA. And they have a protein coat surrounding that nucleic acid, and that typically is very useful for classification. And the other thing is that some viruses, not all, but some viruses are also, the entire protein coat nucleic acid assembly is enclosed inside a membranous envelope. The envelope, in fact, is a phospholipid membrane. It's derived, it's not made by the virus itself, but is derived from the cell that it infected. The virus buds out of the infected cell and takes a bit of the membrane with it. So it's surrounded by a phospholipid membrane. We'll talk more about that later. So every virus has a typical host range, and the host range describes both the type of host and the type of cell that the virus can infect. So plant viruses obviously will infect plants, and it's only certain plants. Animal viruses will infect animals and only certain animals, although that can change with time as they change, and you get exposure of other animals to a particular virus. It may be able to colonize it. In fact, it's very common for predators to be able to be infected by the viruses that are typically in their prey. So in a predator-prey relationship, often you get the viruses that are in the prey also being able to infect the predator. So host range is important to consider, whether it's animal, plant, fungal, protist, protozoal, or bacterial phage, bacterial viruses. And also there are subcategories of cells. Not every cell type necessarily will be in a host can be infected. For example, the influenza virus that infects epithelial cells in the lungs in humans is not going to infect your liver. You won't get influenza of the pancreas. It's only certain cell types can get infected, so there is also a cell range. And typically that range, both host and cell, is determined by what attachment sites are available on a host, whether the virus can attach to the host cell or not. In the case of viruses that infect bacteria, they're that small that they're, can land on the surface of a bacteria, and many can land on the surface of a bacteria and infect it. Those are not called virus, but they're called phage. So those are called bacteriophages. We have animal viruses, plant virus, fungal virus, but for bacteria it's called phage, a bacteriophage. Here's a diagram showing relative size. This is, just to give you some idea, this is an E. coli bacterium, 3,000 by 1,000 nanometers, or 3 by 1 micrometer. Human red cell, usually 8 to 10 micrometers, 8,000 to 10,000 nanometers in diameter. And the viruses, most are much smaller. The Ebola virus actually is quite a large virus. It's one of these long extended structures. There's a bacteriophage. Looks like a lunar lander, kind of. That's a T4. It's very commonly used in the lab. Okay, let's move on. The overall structure of a virus is that a completely assembled infectious virus or viral particle is called a virion, and that complete virus contains nucleic acid, which, as I said earlier, could be either DNA or RNA, depending on what virus you're looking at. The other thing is that it'll be either single-stranded or double-stranded, and it may be linear in form or circular. These are typical things that are used to categorize viruses, whether it's DNA or RNA, single-stranded or double-stranded nucleic acid, and linear or circular. Those three things are key identifying characteristics. The protein coat around the virus, around the virus's nucleic acid, is also important in identification. It's a protein coat called a capsid. It is made up of one or more capsomeres. The capsomeres are protein subunits that make up that capsid. The arrangement of capsomeres around the nucleic acid to form a certain structure around the nucleic acid on the inside is characteristic for each different species or type of virus, and so is also used in identifying it. And as I said, some viruses will have an envelope. It is derived from the host cell membrane. That word is missing here. There should be host cell membrane. It is host cell in origin. It's a phospholipid membrane that some viruses can pick up as they exit their host cell. Once they took over the host cell machinery and started replicating themselves, and they bud out and they take some of the host cell membrane with them, it quite often contains viral proteins embedded in the phospholipid of the envelope. In general, in terms of general shape or morphology, some are helical, a long rod with nucleic acid inside that long cylinder. The capsid forms a long protein cylinder around the nucleic acid on the inside, and an example of that would be Ebola. Or they could be polyhedral in many faceted shapes, like a crystal almost. Some are complex, and bacteriophage would be a good example of a complex one. And some of them, and it could be any of the shapes above, may have an envelope around them, so they may be enveloped. Here again, this is a helical example of a helical virus. This is Ebola. This is a short, what they're showing here in the diagram here is a short section cut from that. So the artist is just showing this area in here. And what you have is, you see some of the nucleic acid inside it, and you see the capsid or protein outer protein capsid or protein coat made up of individual capsomeres. They're repeating units of capsomeres, and they can actually form into crystal-like structures, which you'll see in a minute. Polyhedral ones are multi-side geometric shape, very much like crystals. Many, not all, are what are called icosahedral in shape. 20 faces, 12 corners. And what is meant by that is this. So here you can see that it has these faces, these flat, planar, triangular shapes, and there are 20 of them, and there are 12 corners. Each one of these is a corner. There's a corner. Whoops. There's a corner right there. Corner there. So it adds up to 12 there. Adds up to 20 faces and 12. This is a icosahedral shape. The electron micrograph shown on the right side of the slide here is showing the structure, and you can even see the round capsomeres in this electron micrograph. Okay. The next general type of morphological shape that some other ones are complex, and as I told you earlier, polyhedral, bacteriophages, the bacterial viruses that infect bacteria are in fact complex. They have a polyhedral head and helical tail. So the head is polyhedral and the tail is helical, and those two form the overall capsid. And a diagram of that is shown here. This is showing the polyhedral head with faces and corners, very much like a crystal, and the nucleic acid is inside. And then here you have the polyhedral section, not the the coil, the long extended section. Somehow that word's escaped me, and I can't go back on these slides. So here, what you see in this falsely colored, that's artificially colored electron micrograph, which is a photograph taken with an electron microscope, are 3 bacteriophages on the surface of a bacteria. And what you also see in green is the nucleic acid material, and it's actually being injected in, and you can see it just entering in to the bacteria. So helical. So the complex ones are part polyhedral and part helical, and that's what a bacteriophage looks like. So it's a landing on the surface here of the bacterium. That's the complex shape. As I said, the envelope is found on some bacteria, surrounds the capsid, roughly spherical, and dotted with proteins coded for by the virus. And an excellent example of that would be influenza virus. As you know, there are many varieties of influenza virus, which is why you need to get vaccinated every year to the new varieties that crop up each year, substrains of influenza virus. It is a helical virus. As you can see, it has a long, the capsid forms a long helical, you can't see it all, but it's curling around like this inside. So that's a longitudinal section through it, showing the interior nucleic acid coiled interior. I don't know if coiled would be the exact right word, but the nucleic acid inside. And surrounding all of that is an envelope. In the case of influenza virus, you have an envelope. And the spikes on the surface of the envelope are viral proteins coded for by the virus. And here's an electron micrograph of that, and you can see the roughly spherical envelope and the spikes poking up at the surface, all over the surface. You have these spike proteins, viral proteins, okay, everywhere. That's influenza virus. Okay, in terms of nomenclature, every organism has a genus and species name. Escherichia coli, where Escherichia is the genus, coli is the species, that's for bacteria. Or Staphylococcus aureus, genus Staphylococcus, species aureus. Or humans, Homo sapiens, Homo is the genus, sapiens the species. Some organisms have a subspecies or variant designation. And if we go up in the opposite direction, more general groupings, different, and this is the plural for genus, genera, several genera can be grouped together into a family. So if you have Staphylococcus aureus or Staphylococcus pyogenes or Staphylococcus epidermidis, they all fall under a family. They're all in the same genus and they all fall under one family. Family names for viruses will end in viridae. So it's the name and viridae at the end. Genus names typically will end in virus. So you have influenza virus. Species will have a particular name that's not usually necessarily Latinized. It often is. They'll also have common names, which makes it a little difficult. Names that the public out there use, but not necessarily scientists, and certainly not in an official designation. Subspecies often are not always, but often are designated by a number. And let's just look at some examples of that. For example, if we were to look at these two examples, I'll split it in half. If we were to look at human herpes virus 1, okay, this is belongs to the genus herpes virus, and the species is HHV1. It has several different member species, so there's herpes. Human herpes virus 1, human herpes virus 2, human herpes virus 3. We'll go through these actually. HHV1 and HHV2 are the things that cause cold sores. HHV1 more commonly, but not necessarily restricted to around the mouth, and HHV2 usually genital, although it can be around the mouth also. So human herpes virus 1, human herpes virus 2. These have also older names like herpes simplex virus 1, herpes simplex virus 2, but the proper, more modern designation is human herpes virus 1, human herpes virus 2. This belongs to the genus herpes virus and the family herpes viridae. There's that ending, viridae. Here's another family, Retroviridae. A genus in that family is lentivirus, and an example of a species of virus within the lentivirus genus is HIV-1. There is also another species of human immunodeficiency virus also causing AIDS, which is HIV-2. So we have two species of HIV virus, 1 and 2, in the genus lentivirus in the family Retroviridae. These are large tables, several tables going through different viral families, showing examples. We're going to learn more about some of them, not all of them. I wouldn't want you to try to memorize all of this. You may find it useful later. You may want to refer to it a little bit. I'm going to just highlight the ones that I know that we'll talk about. We will talk about papillomavirus. This is a virus that causes warts. Human papillomavirus, not human wart virus, human papillomavirus, HPV. All of you, in fact, should have been vaccinated with HPV vaccine. The human herpes virus, HHV1, that I just discussed, which includes the older name human herpes simplex virus. So it's called sometimes simplex virus. We'll talk about that later more. The varicella virus, which is human herpes virus 3, that causes chicken pox and shingles. What else will we talk about? We will talk about this later. Rhinoviruses, which cause the common cold. Most cases of common cold are due to rhinovirus, not all. Coronavirus, and there are several species within coronavirus that are important, including the coronavirus species that causes SARS, and the coronavirus species that causes MERS, the coronavirus species that causes COVID-19. All of those are the names of the diseases, not names of the virus species themselves. These are all the genus names, so important genera. We'll talk about that. What else? The influenza virus, it belongs to a family, happens to be called orthomyxoviruses, well, orthomyxoviridae. Remember, viridae indicates that it's a viral family name. All of these end in viridae. Influenza virus, there are three major types, A, B, and C. A is the type that is most important in terms of vaccinating, because it's the cause of most most transmissible flu cases are type A, although type B is also important. And the vaccines that we have each year usually contain at least a couple of different type A and one type B. We'll talk about that more later. We'll talk a little bit about oncoviruses. Let's move on. I don't need you trying to memorize big tables like that. Okay, viruses can be grown in the lab if they are bacteriophages, viruses that infect bacteria. Then what you do is you plate a lawn of bacteria. A bacterial lawn refers to a petri dish that has been inoculated over its entire surface. So you can pour a solution of bacteria over the surface of the agar, the nutrient agar in the dish, and spread it around. It'll get absorbed in. And then you can put on your sample of bacteriophages. Wherever a bacteriophage lands on that lawn of bacteria, that'll appear the next, the bacteria don't appear the next instantly when you put them on. You have to put it in the incubator, and they'll grow as a confluent collection of colonies that are all touching each other. So it forms a continuous lawn, what's called a lawn, across the whole surface. And wherever there was a bacteriophage, wherever a virus was, you will get a hole in the bacterial lawn, and those holes are called plaque. So this is a photograph of the surface of a close-up on the surface of a petri dish, where you have these holes in the bacterial colonies that are covering the rest of the plate, plaques. Wherever there's a plaque, you had a virus that infected the bacteria, took over the bacteria, a bacteriophage, infected bacteria, took it over, made more, and they came out and infected all their surrounding cells and lysed those bacteria. So you get a plaque. It's a little, that's for bacteriophages, and they're easily seen. It's a little more subtle for other types of viruses that infect plants or animals. They grow in living cells and often, but not always, lyse the host cell. If it lyses the host cell, then you can see it. But often there isn't a lysis, but there's what's called a cytopathic effect. So you see damaged or abnormal cells rather than lysed cells. Lysed cells are easy to spot because there's going to be a hole in the culture. If you have a lot of cells and there were some viruses on top of that, then that could lyse the cells, then you'll see a hole. But otherwise, quite often with animal viruses, you get a cytopathic effect. So you have an area that has abnormal cells. Animal or plant viruses have to be grown in vitro in a dish in the lab using cell lines. And what you do is you have the cell type that they can infect and you add it on so it can be continually grown in culture in the lab, and they will cause some sort of change so that cells are transformed. You see the CPE or cytopathic effect. Here is an example. These are normal fibroblasts growing in a nice stretched out manner, and here you see these cells have been infected with, I'm not sure which virus, but they have been infected with the virus, and they are rounding up and not as attached or spread out on the surface of the plastic. Another way of growing cultures, not in a dish, a plastic dish, in individual cells, but in vivo, which means in a living organism. So the third way is to grow them in vivo, and here we use eggs quite often because you need a living animal, certain tissues, and there are different areas in the egg that can be inoculated. And people who do this kind of work are really specialized and really skilled at getting the needle just into the right area of the egg, chicken eggs in this case, to grow the virus. And a lot of vaccines actually are prepared in eggs, in chicken eggs, which is why they would ask you when you go to get vaccinated, if it was prepared that way, they would ask you, are you allergic to anything? Are you allergic to chicken or eggs? And it's because the virus was prepared and grown in an egg, and then purified from that and killed so that it could be vaccinated into you. So, in order to identify a virus, we look at its cytopathic effects. Sometimes we use serological tests looking for antibody. If you can detect the antibody in a patient, that means that that person was infected and did respond. I mean, with an infection, typically you do get antibodies forming. And you can find them, detect them in the patient's serum. Sometimes we use purified, highly processed antibodies for identifying viruses. Sometimes it's different kinds of tests that allow us to identify what virus is present in a sample. Another way of identification is to look at the nucleic acids. I'm not going to get into these techniques, but they are very specific. These are the kind of things that are used to see if someone is carrying the virus. So using PCR, which happens, I wouldn't worry about it, but it happens to stand for polymerase chain reaction, you can determine if the nucleic acid for a particular virus is in a sample from a person. So then you know, well, they have the virus. When you detect antibody, well, that's after the fact. The person had the virus and they responded by, their immune system responded by producing antibody. So that allows you to, if you test for that, it allows you to find out if the person was exposed, had been infected to the, had a viral infection and got over it. The PCR would be a way of telling if the person had the present case. Okay, in the case of bacteriophages, we'll do bacteriophages, then we'll do animal viruses. We'll talk about how they multiply in their host cell. And I think at this point, I just want to stop and make this the end of Lecture 7, Part 1.